Ideally, we would have like an intro to CLS at the beginning of this program. We don't have that yet, but safety is usually something that's covered in that. By this part of the program, you should be pretty familiar with safety. But just keep in mind, hand washing, PPE, sharps containers, bio trash, like proper things in the lab, no food, no applying makeup, keeping your hair up. In this class, any glass tubes need to go in a sharps bin. Anything that's not glass can go, you pretty much only have sharps bins in here. Unsoiled gloves can go in the regular trash. If it has blood on it, then it needs to go in a bio bin. Any questions about safety? You're also going to have a professionalism grade thrown in here. So this is really tiny, but it's posted in the course. So these are things to keep in mind, like cleaning up after yourself, being prepared for class, following safety rules, a lot of things. So centrifuge, for these centrifuges, these are really, really old centrifuges. So they're not the greatest. But generally in the blood bank lab, we do an immediate spin read, which is you just mix your reagents and then spin for 20 seconds. Most of these are programmed already to automatically spin at 3,500 RPM. But sometimes when you take them out, it makes you reprogram it. So 3,500 is what we will spin at during this class. So if you're doing an immediate spin read, it's 20 seconds. If you're washing your cells, it's a 60-second spin. And is everyone familiar with how to balance a centrifuge? Okay. So documentation. A big thing about blood banking, because you're reading a lot of tubes, is concurrent documentation. So as soon as you pull a tube out and read the reaction, you need to document it right then. Because if you don't, and you're just relying on your memory, that's not good practice. And pretty much any SOP you read in a hospital will say concurrent documentation is required. You need to use indelible ink, so no pencil should be used in this class. It should be black or blue ink. So error corrections, never scribble out errors because it makes it look like you're hiding something and you don't know what it was that you wrote under that. So the proper way to error correct is just one line through it, date and initial. So if you document something incorrectly, just strike through, initial date, and then write your correction. And one big thing, antigens versus antibodies. In this class, if you are talking about an antigen, you're just writing whatever the name of the antigen is. So if I write this, JKA antigen. If I'm talking about an antibody to JKA, you have to either write anti hyphen in front of it, or just hyphen is the short form to write antibody. If you do not write this or this, it's assumed that you are talking about the antigen. So on exams, when I say what antibody is present, and you write this, if it's really this is wrong. Does everyone understand that? It's extremely important to say the right terminology in blood banking so that these workups are often very long, so they can be longer than 8 hours. So you might start a workup and not finish it. So if you pass something on to the next coworker and you say this patient has a Duffy A, and you really mean an anti-Duffy A, it could make them do extra work that's not needed. It could make them issue the wrong unit. So terminology is extremely important. And this class is intense. It moves very quickly. There's a lot of information. There's a lot of extra credit. So if you don't do well on the midterm, don't worry, there's tons of extra credit. You should all know statistically that clinical labs help with the diagnosis of between 70 and 80% of diagnosis is based on labs. So our job is very important. So I want everyone to know that. And take your job seriously, because what you can do if you do something wrong, it can be harmful to a patient. And just keep that in mind when you're working in a lab, that every test you do is affecting someone. Any questions before I move on to ABO?
Is the lab the same grade as the lecture or something?
It's separate. Okay, so talking about ABO. So it's all based on Landsteiner's laws. So the first one is if an antigen is absent on the red cells of a person's blood, the corresponding antibody must be present in the plasma. This is only true for the ABO system because these antibodies are naturally occurring. But the second law, if an antigen is present, then the antibody must be absent. So this is true for all blood groups, and it's an important thing to remember when you're doing antibody IDs. Basically, you shouldn't be making an antibody to something that you have in your body. Your body should recognize it as part of you and not mount an immune response to it. So going back to the first law, if you are group A, that means you have A antigen on your red cells, and it means you should have anti-B in your plasma. You should make the antibody against the antigens that you are negative for. So if your blood group AB, you have both antigens, so you won't make any antibodies. And if you're group O, you don't have either A or B, so you should make both A and B antibodies. Does this concept make sense to everyone? So this is just the chart of what you should expect. If you have one of these blood groups, this is what your reactions will be when we're testing. So after we test today, you can come back to this chart. So the reason I grouped all the carbohydrate antigens in one module, even though they seemingly don't have that much in common, they are all carbohydrates, which means they're not direct gene products. So when something says, there will be trick questions like the A gene makes the A antigen. True or false? That's false. The A gene does not translate for the A antigen like most of the blood group systems. For all the carbohydrate antigens, the gene translates to a transferase that adds a sugar to an existing carbohydrate chain that makes that antigen. So just keep that in mind. They are not direct. Any of the carbohydrate antigens are not direct gene products. They're not making the antigen, they're making an enzyme. So in line with that, you have to have the H antigen to be able to have ABO antigens. So the FUT1 gene makes this transferase, which adds a fucose to a type 2 structure. Once that fucose is added, that is the H antigen. If you have the H antigen, if you have A or B genes, then they will add either if you have A, it'll add, and I just say the abbreviations, it's easier to memorize everything. So GalNAc, which is N-acetylgalactosamine, makes your A, and then B adds a galactose. But you must have the H gene. If you do not have the H gene, you'll have the Bombay phenotype. So even if you have an A or B gene, you will not produce those antigens because you have to have H present to add to. And these are just different pictures to show. Adding that GalNAc to make the A, adding that galactose to make a B antigen. Any questions? Okay. So I'm just going to throw in some advice throughout the labs. So if you understand what's happening in whatever you're doing, it's going to help you memorize everything, and it's going to help you be able to troubleshoot when things go wrong. So if you understand right now, I'm washing the plasma away from the red cells, then you can troubleshoot later. Just keep in mind, every step you do, try to think about what's being done. What's the purpose of this step? So first, what we're going to do is just do a blood type. So So when you get a tube of whole blood, you will spin it down. These should already be spun down, so you don't need to spin it down. And what you want to do is take 2 tubes and label them the patient name. And you'll have one for RBCs and one for plasma. So today, your patient name is really your donor ID. So your last 6 digits on your tube, you can just use that. And then I was going to explain donor IDs as well. Okay. So you all have donor samples. When you get a donor ID, so this is for a blood unit, it will have a DIN on it. So that's donor identification number. The first little section is the facility ID. Any FDA approved facility for collection of blood products will be assigned a facility ID. So you can always know, if you look at a blood product, you can look up who it's from. So this, for example, is Life South. If you get one from Stanford, it would be 05 or 0750 is one. Some blood centers have multiple IDs, but there is a database. So if you need to look up where something is from, you can put that in and it'll say this was collected at Kentucky Blood Center, the blood center of the Ozarks or whatever. So this is the facility ID. This is the year it was collected. This is a unique number. This is a DIN flag. So some blood centers do not use DIN flags. Basically, when you collect blood, there will be a bunch of stickers on it. So some stickers will go onto the unit itself. Some will go onto the tubes that will go to the donor testing lab for disease testing. And some will stay on the bag. Be used on paperwork or whatever you need to use it on. So for centers that use DIN flags, every single sticker on that DIN pad will have a different ID or DIN flag.
What's the first word you're saying? The kind of flag?
DIN,
EIN?
donor
DIN.
identification number.
Okay, thank you.
This is the check digit. Which this is, there's a calculation that adds all these together and then shoots out a check digit. Some donor centers do not use the check digit either, they just use this number. But basically, these are meaningful. These will be unique. So on your paperwork, when you're working with a donor tube, you can use this part of the DIN flag as your patient ID or your donor ID. So you'll lay it go along 2 glass tubes with your donor ID, and one will say RBC and one will say plasma. You'll use a transfer pipette to pull all the plasma off your tube and put it in the plasma tube. You can do things as I'm saying them. When what? You didn't get a sample? Okay. So after you have pulled off all the plasma, you'll take 2 drops of the patient RBCs and put them in the RBC tube. And then when you do washes, you'll want to fill the tube 3/4 full with saline. And you'll see there's some other bottles on the benches. There's VI water, there's ethanol. Be very careful not to select the wrong thing to wash your cells with. So if you use VI water, it's going to lyse all your cells. And you're not going to have any cells after you wash. So if you have anything near you that looks like the saline bottles, I would move it out of your way so you don't accidentally kill all your cells.
The no saline water or yeah? Yes.
Yes, only saline. So you're gonna fill it 3/4 full and wash your cells. So after you have 2 drops of RBCs, 3/4 full. And then you're gonna spin for 1 minute. So you can use a balance tube, just full of saline and that's it. So you're gonna put all the plasma onto. Oh, all of. And. We didn't need. We didn't. We don't have a. And someone can use the rest, people know in there. So we don't have. Some of you are going to get it up. And there's. So the other thing to keep in mind, you have supply shortages all the time, so if you can reuse a transfer pipette, you don't have to throw it away every time you drop something. It's wasteful and there are transfer pipette shortages. So if you're using that transfer pipette for the same thing, it's okay to. So you can get one transfer pipette in your class for the whole time. And then for your red cells, so after you add your saline, you want to make sure that you mix it up. Because there's some stuff on the bottom are not getting washed. You just want to make sure that you wash. So the purpose of this is to wash any plasma off your RBCs. So, and you can spend it for 1 minute. Right? Yeah. Well, we're not doing this. So the other thing you want to make sure whenever you're using like a sailing ball, never want to stick the stem into the tube. So always make sure when you're dropping something or you're adding saline, you're squirting from above the tube. So after you add your 3/4 full, that's what way you want to do that. Yeah. So 60 and then 2 drops.
Yeah, but I think maybe a little more.
So it's best my close to other focus on the same time. But like if you're experiencing like or automation, they're not wrong. Most patients is not like going to cause but they're full antibody and if you it just saves the step. If you do it in the beginning, the initial is 20 seconds. So when you get your sample out, you can put dump into the bio bin. Yeah. So that's why you have to do 1 minute so that your cells are packed enough, they're not on it. So. Yeah. When you're making a 3% suspension, you're basically going to add some saline and then compare it to your reagent red cells. They're the color. Yeah, you can take it out. That minute. So if it's a little bit lighter, or a little bit heavier, it's fine. But this is a big one. That's what we're doing, right? So before it says, okay. Some of these. We've had some issues. Yeah, when we put it in, it looks like. That was. What are you talking to? Yeah, we might, I don't think we put in the ditch under. I think they said. Yeah. You see that? And this is. I did it before. Good job. But if it works. When it gets it should be on, I'll bring that. You don't have to do it. And then. I can all. If it's too heavy and you need to take on the storage, so you can have nothing. And then you said we just pour this right in the. So as you get familiar with the process, keep in mind when you can do things. So if you're spinning for a minute, you can be thinking about what the next step is, and you can start labeling all your tubes for the next step to save time.
How do you put the time or the speed?
Our hands should already be set.
Okay, so I put it. Okay.
There's one in here. It's yeah, it's on the AGO typing. Actually good on page 7. Yeah. So is it like 3/4 again? Yeah, that's what I was thinking, but it's. Where are those? That one.
Water, right? No, I meant, don't you like? I'll take it. Yeah. Yeah. I can. That's probably, yeah. Hey, did you draw them down right? Dark. Wanna put something else. I think that's not a even I was saying. That. They might be dark. One have there just like working on it. Once everyone has. Oh, so you wanna do it quickly? Or you're gonna download yourself? You do it slowly and drag out. Oh, yeah. 3 minutes, so you can start that after. And hey, can we open this? Make sure when we're preparing to read that it's well mixed. So it goes on. And you want to make sure you're pressing the rock so that if there's any cells or fluid stuff in there, that they're being continued. Okay, everyone that has their 3% suspension can start labeling their tubes. So you're gonna have an anti-A, an anti-D, anti-D, and then an A1, and a B. Do we put that whole number on each also? You don't have to if you're working with one sample. Okay. If you're working with multiple samples, then you want to label it something to. We're labeling the two. So then, yeah, you're also, we're gonna have screen as well, so that they can start incubating. So you're gonna have S1, S2, S3, and AC, which is your auto control.
Way more, use the, oh, so that everything's mixed together. I don't say that, and then mix it up. Layer the bottom. The first one is. Okay. So you always want to drop your clear liquids first. So you can drop your anti-A, your anti-B, your anti-B. That's the anti-A. Did everyone drop their anacera? Anti-A, anti-B, anti-B. So your reagent rack should be set up so that the front row is anti-A, anti-B, anti-B, A1 cell, and B cells. Should be your front row of your reagent rack.
I need more NPA. I cannot pull it out.
Okay, for your reverse and your screen, these are all testing your patient plasma. So you're going to add 2 drops to all of those tubes. 2 drops of patient plasma.
Before you add the reagent?
You always add your clear liquids first.
Okay, so the reagent's going first and then.
Well, not for your reverse. And your reverse, your clear liquid is your patient plasma.
Oh, the plasma, okay.
A1 and B.
I think you do. How dirty you get.
And the screen, yeah.
Okay, does everyone have anacera and plasma drop?
Oh, no.
Yeah. How many of the plasma? I got questions.
2 drops. All the instructions are on the PowerPoint.
So just do your ADO, so you do reversal. And then. Plasma and drop. Yes. Plasma. Yeah, you drop. Okay, so after we drop our anisura and our plasma, all of our clear liquids are dropped. So the reason we drop all of our clear liquids is after you do that, you want to look at your tubes and make sure all your tubes have something in them. To make sure that you didn't skip a tube, forget plasma, forget anisura. All your tubes have something in them. So you confirm that? Okay, so now we're going to add the red cells. So in your forward, you're typing the patient cells. So you're going to add 1 drop of your patient suspension to your forward tubes. You're also going to add 1 drop to your auto control. Your auto control is patient plasma plus patient RBCs to see if there's any auto reactions going on. Auto control. So you've added your 3% suspension to 4 tubes. Yes. Okay. Then your reverse is you can add your A1 reagent cells to your A1 tube, your B reagent cells to your B tube, and then we're going to have to share screen cells. Once you drop them, so make a note when you get the screen cells, what lot number it is, because you will want to record your reactions on the anagrams for that lot. What is AC? Your auto control. So that's patient plasma and patient red cells. You should have for down plasma and plasma is 2 drops. Everything that's plasma 2 drops. And red cells are 1 drop for everything. Wasn't supposed to add that to the anyway. Blood, right? To the A1, the NAA, yeah, what A1, and then the also, and then I have the money, but then that's the AC, right? Not the F. And that's where you go. So to record the ABO, you will use this ABO worksheet. And you're going to be doing multiple donors. So you'll just record your donor ID for your patient or donor ID here. And when we start reading the reactions, we'll record the reactions here. So just get 1 from each stack. And then when you get the screen cells, you have screen 1, 2, and 3. Drop 1 drop and each and pass the screen along.
So the vials on the screen cell will have a lot number and expiration. So you got 3 sets of anagrams. So you want to find the lot number for the vials that you get when they're passed around. So go ahead, once you drop 1, pass it. Once you drop 2, pass it. You don't have to pass the whole group together.
Should be getting cooler really soon.
Okay.
Okay.
Screen 1. So there's screen 1, 2, and 3. So just make sure you're dropping the right cell.
It's coming around. All right, now I added my OneDrive, so I still have to do the A1 and B. Yes.
Okay, when everyone is dropping the
Yeah,
cells,
that's what I'm saying, I need to
we're gonna talk about next steps.
Yes.
So after you've added everything, you want to give your tubes a quick mix to make sure your cells and reagents are mixing together. So you can just do that with your wrist, just flip the tubes. When you add your tubes to the centrifuge, so you're just dropping 1 and 1, 2 and 2, 3 and 3.
I can't get it to come out of this.
Screen 1, screen 2, screen 3. So you drop the patient cells. This one, and this, and this. And don't drop this one. So you just need to do these. They're not the right ones. You want to make sure you have the right one.
So it's just the plasma and the red blood cell, right? Yeah. So I had plasma and I added red blood cells, so that's all I added, right? Yeah, but I had that in there, obviously. Plasma and, yeah, the red blood cells. But there's no reagent, right?
So the screens are being passed around, so you don't have the.
No, I meant the auto, it's just the plasma and the red blood cells,
Yeah,
right?
you're just testing for autoantibodies, so you
Yeah,
just have
so
to.
that, I don't have another reagent, okay. That number, right?
Anyone that screens, it's not gonna meet the anagram.
Right. Oh, yeah. That's what I was just saying, that was our lot, we were working on many of those papers.
Have one.
So if you use the screens in your rack, it's fine. There should be a negative screen anyway. But the reason we're passing around the screen cells are because they're the freshest. So once cells degrade, they're not going to have great reaction. Some antigens degrade over time. So that's why we were passing around the freshest ones. So once you've dropped your screen cells, you're going to load all your tubes into your centrifuge. And you want to develop a practice for yourself to always do things in the same order. So ABO is the most important test in blood banking. It is the easiest way to kill someone by making a mistake. So you always want to load your tubes in the same order. You always want to unload your tubes in the same order, so that you have systems set up so that if you get a call from a hospital in the middle of reading tubes, you can go right back to where you left off and not get confused. So now after mixing your tubes, just lightly flicking them to make sure they're mixed, put them in the centrifuge, and you're going to spin for 20 seconds.
Yeah. Put it into those. So we want to have everything has fun. Yes. And we'll talk about why the lot number is important while we're incubating, because we have a 15-minute incubation time. So all of your tubes should look red, like they have red cells in them. So you want to give it a flick with your wrist to mix everything up and then spin. So I would recommend having your tubes in this order so that when you pull them out, you can read them in the right direction. So before you start reading, let me come around and watch you read. Reading is one of the more difficult parts of blood banking. You just have to get familiar with what reactions look like. Huh? You already read? How long? Hey, you can do whatever you want. It's okay. Whatever's in your right.
Yeah, yeah.
It's negative. It should work. So basically, one of these things, until all the songs are used in. Because that's really the very on the side. In the business. So, what you're looking for. And so it's all when you just go over there and don't get out of this. And then you. So remember, we're on current documentation, so we're taking out one tube at a time, looking at it, reading, documenting that here. Before we go on doing that. You're. Don't really. And the extra logo. One big thing for it. So when you're reading your reaction in your elimination viewer, you're not really looking at your tube, you're looking at the mirror. So you want to angle the mirror in a way that when you shake your tube, you're looking at the reaction in the mirror. So before shaking your tube, you want to bring your tube over the mirror. And you're just gonna tilt it to see if your scalpel is going to dislodge at all. So that's called looking for the tail. So if you have a very negative reaction, you should, or you might have some cells start trailing down. If you see that, then you can lightly start clicking. So if it's negative, all your cells are gonna break up. And you're not gonna have any constant on. It's gonna be just like the way it looks in your suspension. That's negative, no elimination. Above, you're always looking for elimination. So if it's 4+, everything's gonna come off in one chunk. If you have one chunk, 4+ reaction, all your cells are in one big clump. And then the grading goes down from there. So if you have multiple large clumps, that's a 3+. If it's a bunch of smaller clumps, 2+. If it's really little clumps, then it's 1+. And you kind of just have to get used to it.
What happens if you have a couple of little chunks and then you also have a tail? Is that?
So there's mixed field reaction, which reading mixed field reaction. So one concept that's really important that everyone gets confused about a lot is mixed field reactions. So you have mixed field reactions when you have two cell populations. So that can be, if I just had a transfusion, I have my own cells and I also have donor cells. So if whatever I'm testing against anti-A, if I'm A, but I was given O cells, I have A cells and O cells. So those O cells are not going to react with the anti-A, but my cells will. So the O cells will give you a negative reaction, so you'll have some suspension. But then most of my A cells will be in a chunk because they're eliminated. So the main takeaway, if you see mixed field reactions, you have two populations of cells. That's what causes mixed field reaction.
But for what we're doing right now, we shouldn't be seeing that, right?
No, you don't know.
So it's
Yeah. So in mixed field is a lot easier to see in gel than it is in really.
I could be wrong. I think, have you read the other one yet?
And then we'll talk about recording reactions too, in a little bit. In blood banking, we don't really record negative. We always record 0. So after we get done reading the cells, we will add 2 drops of an enhancement. So in your rack, you should either have PEG or LIS. And so PEG, there's gamma PEG, and then LIS, one of the brands is called MUAD, but it says low ionic strength medium, which is what LIS stands for. You can add either one, that's 2 drops. To the cell, just a screen. These are the forward type. This is the reverse type. This is the screen. So the ABL, after you read the ABL, they can be discarded in the charts. But the screen, so screen 1, screen 2, screen 3, and auto control. After you read them, you're going to add 2 drops of PEG or LIS. And then you're going to incubate at 37. So there's a 37-degree incubator here, or Or on the sides of the gel incubators, they also have tube holders. So these two are on. Okay.
My SID has a real good spot. I said that was a 4.
So that one, because it has a tiny little satellite, we call it 3+. If it's not in one solid, then it would be 3+. If you have a big pellet, but you have some little mini kind of agglutinates off of it, that would be a 3+. 4+ has one pellet. That's it.
Now, A1.
So, yeah, that's weird, because it does want to make it. It looks like there's some sounds.
And it's the same on the B too. B also.
What would we call that? Let's see. I think I would call that a 4+. I think it's just because it's been like chicken so much that one little piece fell off, but that's a pretty solid 4+.
So the B one of 4+, and then the A.
Yeah, the A is breaking up, but you really want to read by this. So, yeah, so I think it would have been a 4+. But I mean, if you just keep shaking and shaking, and then you're straining. So you're go to these away, okay, and then the strains, we're gonna have 2 drops of this, okay. Yeah. And 2 drops of each to the, yep. So the auto control, you wanna do whatever you're doing to your strain, to your.
Oh, yeah.
I'll just put it in the back.
The screen you want to record on your hand. So. It could be that one. If you use the one with the rack.
Oh, right, I'm like, you don't need it, right?
Is it 1 pump? Yeah. Right. But I want to record your patient review before you report any reaction. Oh. Yeah. One part of that. Yeah, I just have a board, but it's not
Yeah, now they're just saying. She did say the longer you wait. Yeah, the oil, yeah. But this
Okay, so you're incubating at 37 for 15 minutes. Yes.
So I started timer, right?
Yeah, we have an incubator.
What you need to.
No,
Everyone needs to report on like I said, if you're logging your RH results on the RH, or when you're reporting your stream results on the Amazon, so that's good.
Oh, yeah.
Yeah, not it.
Do you remember what it was like?
It was, I thought it was about a 3 maybe people who were, but they were just.
Oh, no. I thought I have yours. I do. I already spun mine. I'm hoping I can spin mine again. So that you can see that you want to. Question I want to ask you. Because you made us so long, so that then the A1, A2, well, the A1 and the B sections were on like this. Would it be okay to put back in spin it so that I can see what it looks like? But we don't do that normally in the lab, right? Okay, got it. Let me share this.
you're incubating your screen.
Yeah, it's over there now.
Yes, when I'm in the this evening. I would start writing my IBIS ID. So we're going to talk about what we got. So the thing that you need to be very clear on is what is happening. So your forward type, you are looking at what antigens are on your patient cells. So you have this known antisera that you're using. So anti-A is always blue, anti-B is always yellow. That is an FDA rule. Because blood banking is so important to patient safety, FDA has mandated that any anti-A and anti-B must be this color to prevent mistakes. So that's how important this is. So you want to know forward type is looking at what antigens are present. So this is what's telling you what your ABO is. So if you have, what was your reactions for anti-A and anti-B? Okay, so what does that mean? That means that that donor does not have A or B antigens. So if you do not have A or B, that makes you an O. Okay, so you want to make sure that your reverse type matches your forward type. So going back to Landsteiner's law, if the antigen is present or absent, you must have the antibody in the plasma. So if you have an O, you must have anti-A and anti-B in your reverse type. So what reaction should you have for an A? Or sorry, for an O? Yes, 4+ in both of your tubes. So these are reagent cells that are known to be A1 positive or B positive. So it's telling you what antibodies your patient has. So those two must match up. So if they do not match, that is called an ABO discrepancy. Everyone clear on that? So what is an ABO discrepancy? Correct. So that doesn't mean there's something wrong. It doesn't mean there's a testing error or you dropped the wrong antisera. It just means that you need to investigate to see what's causing the discrepancy between the forward and the reverse match. And the type of discrepancies that you have are either missing reactions in the forward, extra reactions in the forward, or missing reactions in the reverse and extra reactions in the reverse. So depending on what kind of discrepancy you have, you will troubleshoot and investigate differently. So we already started our screens. So depending on which enhancement material you selected, most hospitals do not do a 37-degree read anymore. Some do, but the thing you have to keep in mind is if you use PEG, you never want to spin at 37 degrees. If you do a 37 degrees, that means you're incubating for 15 minutes at 37, and then you're spinning. For PEG, PEG is so sticky, it can cause a false positive reaction. So you never want to spin PEG after 37 incubation. Instead, if your facility has it in their protocol to do a 37 read, for PEG, you would just pull your tubes out of the incubator, because at this point, after 15 minutes, most of your red cells have settled naturally, and you would look at the plasma and look for hemolysis. So the 37-degree read for PEG would be either H for hemolysis or NH for no hemolysis. But at this point, most hospitals do not even read at 37. They just have an immediate spin read and then a final read, which we'll talk about that too. So what's going on after the incubation? So one thing, why do we incubate at 37 degrees? Yes, thank you. So you want to basically give it a chance if you have any antibodies present to bind to your cells. After your incubation, you're going to wash your tubes. So in here, we don't have cell washers, unfortunately. If you're working in a lab, you'll have a cell washer, but it's good to know how to manually wash anyway, because sometimes if you work in a small lab, you might only have one cell washer and then it breaks and then you have to do manual washes anyway. That would be horrible. But what you're doing is if there's any unbound antibodies, you're washing them out so that all you're left with are your cells. So after you wash, you're going to add anti-IgG, which is so AHG stands for anti-human globulin. So it is literally an antibody to IgG, an antibody to an antibody. So if your patient has IgG antibodies to the red cell, these are your patient IgG. This is your Fab. It's binding to the antigen. So this is your antigen binding region. So your FC region is sticking out off the red cell. You have anti-IgG, and it is another IgG that it's specific to the FC portion of an IgG. It's fine if you incubate over 15 minutes. So if there's any IgG present, your anti-IgG are going to bind to them. So its Fab is binding to the IgG's FC portion. And the reason we need this for IgG antibodies is because IgG antibodies are so tiny. They don't have the ability to make agglutination that we can see because they can really only bind 1 or 2 red cells. But when you add your anti-IgG, it's cross-linking all your RBCs so that you can see agglutination. So this is the purple is your anti-IgG, and it's just giving you a way to see the agglutination. If your patient does not have any antibodies, it won't have any IgG to bind to, so you're not going to have any agglutination. Is this making sense? So after we do that, for any negative reaction, there's one more step, because you want to make sure, because again, making sure your patient doesn't have any antibodies is very critical before issuing blood. So if they have an antibody to a unit, then they can have a hemolytic transfusion reaction, which those can kill people. So we don't want to give a unit that's positive for an antigen that a patient has an antibody to. So to make sure that our test worked, we add these things called check cells. So check cells are RBCs, group O RBCs that are sensitized to IgG. So they have IgG sticking off. So you add those to your negative reaction cells, and you have your anti-IgG, you add your IgG sensitized red cells, and that you should have agglutination afterwards. If you do not have agglutination, that means that either you did not add anti-IgG, there's no anti-IgG in the vial, or that you did not wash your cells adequately. If you didn't wash all, if your patient has a large amount of IgG, it can't bind, everything's not going to be bound to the cell. So it's going to have some free IgG floating around. So if you don't wash all that out, when you add your anti-IgG, basically the Igs are going to bind together and you won't be able to see a positive reaction because it's neutralizing the anti-IgG. So you're getting a false negative. We don't want a false negative because that can harm our patient. So when we add our IgG sensitized cells and we get a positive reaction, that's saying I had AHG, it was not neutralized, it was active, able to agglutinate. Then a grading reaction. So like we talked about, 4+, 1 solid agglutinant. And then it just goes down from there. So 3+ are if you have 1 large agglutinant and then a couple of little small ones, that's still 3+. And then it just goes down from there. If you do notice that your tube looks hemolyzed, that is a meaningful observation. So you do want to note that. So if you're starting with clear plasma, and then after your test, the plasma is now hemolyzed, there's still something going on there that you want to know. Because there are some antibodies that are known to cause hemolysis. So that can point you in a certain direction. Okay. So we're going to talk about antibody screens a little bit more. So the main purpose is to detect clinically significant antibodies prior to transfusion to prevent a transfusion reaction. So that's a part of pre-transfusion testing. You always do an ABO so that you can give compatible units to your patient, and you always do an antibody screen. So another FDA regulation. So you want to detect any clinically significant antibodies. So that's a key part, because there are some antibodies that are not clinically significant. They just cause issues, which can make blood bank testing a little bit more difficult and annoying, basically. But the important ones that can cause transfusion reactions are all of these. So your RH system, your MNS system, your KEL system, Duffy system, KID system, P1, and Lewis antibodies. So every screen that you get should have all of these antigens on them. You must rule out all of these. And this is what an anagram looks like. So for every lot of cells you get, they do make 3-cell and 2-cell screens. And what you're looking at, you want to make sure you're always matching up the lot number with what is on the vial. So up here you have the blood group system, and then below that you have the antigen. So over here, so each vial, some people call each vial vial, some people say cell 1, cell 2, cell 3. It doesn't matter what you call them. But each vial is a different donor. So there's one person in that vial. That's an important thing to know about the screen. If you're looking at vial 1, usually the manufacturer, so this one's Immucor, they'll have a list of donors that will come in and donate a unit of blood, and then they'll make reagent cells out of their blood. So for vial 1, this donor is positive for D, negative for little c, and so on. This is all the reactions for that one vial. And so on for 2 and 3. So each lot, going from lot to lot, is going to have a different donor. A lot of times the donors will be very similar profiles, but they're going to be different donors. So that's why you want to make sure you're looking at the correct lot. So it may only vary one antigen. Like if you look at, this is lot 502. So next month it might be lot 503. The donor may have exactly the same reactions, except Duffy A and Duffy B are switched. So that could cause an issue if your patient has an antibody and you're using the wrong anagram. That can point you in the wrong direction when you're trying to do an antibody ID. So here's an example. There are 2-cell screens available too. They'll only have 2 different cells, but it has the same thing, the blood group system, the antigens, this, and so this one calls it cell, the other one called it vial, doesn't matter. So when we're writing down our results, Our reaction strengths are going to be 4+, 3+, 2+, 1+, or 0. Some people write down an M if they read it on the microscope and there's a tiny bit of agglutination. Some facilities will say don't read on the microscope, so you just have to follow your facility's SOP. And then some people will record a weak reaction. I don't really like that. I like if it's 1+, it's 1+. If it's 0, it's 0. The weak is, it's kind of meaningless. Like you need to make the call. Is it positive or is it negative? And then you want to record what phase you're reading at. So your immediate spin, if you have a 37 read, and for your AHG or IAT, different facilities will write that different ways too. Some people will write IgG, which I don't really like that because it's not really correct. You're really using anti-IgG, not IgG, but you'll see that. AHG, IAT. And then if you do any kind of special testing, so when you do ABO investigations, you might incubate at room temperature or incubate at 4 degrees. So you would record that. So like 4 degrees for 15 minutes or room temp for 30 minutes if you're incubating at any kind of different temperature or time. And then here's some examples. So here's a screen. So your auto control is your patient cells. So you record your auto control on your 4th line. So you have up here what phase you're testing at. So you have immediate spin. And then if you used any enhancement, you would also want to include that. You want all the information here of what you did. So if you use LIS, you'd write LIS AHG or PEG AHG. And then remember, for any negative reaction, you have to test your check cells. So I like to record check cells with just a check in the same box. So basically, if you're doing any tube testing and you have a 0 reaction without check cells, that's an invalid test. So you want to remember that. And then if you do any other testing, you would just document what you're testing. So if you use DTT treated cells, you document that. If you use FISA treated cells, you document that. So as long as you're using the same cells, you can document it all in the same anagram. It's just different treatments or phases that you're documenting. Any questions about how to document?
Okay, so you're saying that for each column, you first redded and like it was 0, 0, and then redded the check cells and added if it matches, you put the check.
So for in the AHG row, yes. So if you read after adding AHG and it's 0, you're going to add your check cells and add a check.
Because it matched that, right?
Well, that's saying that you add the check cells and there's agglutination. So the difference between recording gel results and tube results, in gel, you can't do check cells. Once you spin, that test is done. So when you're documenting on that you're running with gel, you will not see check cells. So you're not expecting that check. So it's perfectly valid. The IgG is in the gel already. So there's not a risk of you didn't add that reagent. It's already in the gel. They have anti-IgG gels specifically for screens or antibody IGs. So I know this is a lot of information. This is all going to be posted later. So, and also in gel, there's no immediate spin testing because after you spin, you've spun it. So for gel, you just add your reagent red cells, add your patient plasma, incubate for 15 minutes, and then spin. So when you're doing gel, you're just going to have one read, which is your final read. And then most people use potentiators or enhancement media into, you don't have to, but it speeds up the reaction. So if you do not use LIS or PEG, usually if you're doing a neat reaction with no enhancement, you have to incubate at least an hour. And nobody got time for that. So that's why we use LIS or PEG. Okay, the other important thing. So different computer systems and different hospitals, they'll have different tests that you can order. So some things are called differently. So they might call, if they just want a blood type, they might call that a type. They might call it ABO-RH. They might call it blood type. So that is your ABO and your D type. So it's very frustrating and misleading that it's called RH type, because it's not your RH group. RH is a system that has like 54 antigens in it. It's just your D type. Is that person positive or negative for D antigen? So it's going to be resulted as A positive, A negative, whatever. You're going to have the ABO and then a plus or minus for the D typing. Or if you have an ABO discrepancy, you're going to resolve it as inconclusive. So this is important because when you take the midterm, you're going to get results and you're going to give an answer. So this is telling you what to answer. So if you get a screen from this, after you do this, and then another thing that's commonly ordered is type and screen. So for type and screen, you're going to get an ABO result, and then you're going to get your screen result, which is going to be negative, positive, or inconclusive. By doing a screen, you're not expected to know what antibody is present. You don't have enough information. You just need to know, is there an antibody? That's what the screen is telling you. Antibody, yes or no. So negative or positive. If it's negative, then you're good to go. You can move on, issue units. If it's positive, then you need to do an antibody ID. So if you do an antibody ID, then you report that as the actual antibody that you find. So if you find an anti-C antibody, you report it as an anti-C. So does everyone understand the difference in reporting of a screen and antibody ID? One of them, you're reporting the actual ID. You're telling them what is present. The screen, you're just saying negative or positive. There is or is not any antibodies present.
So it's qualitative.
Basically, if you want to view it like that. And then if you're doing a cross match, we use different terminology for that. So if you're cross matching a unit that you want to give to the patient, you're going to take some of those cells from the unit and mix it with your patient plasma and see if it's compatible. So the way we report those are compatible or incompatible. You don't report those as negative or positive. For cross matches, you're asking, is it compatible? Any questions? So a type and screen is ordered on a patient. The ABO and D and gel screen are performed, and this is what they got. So look at their forward type. What does their ABO look like?
Say what is the type of because that was
Yes.
The anti-A is 0, anti-B is 0. Anti-D is 4 plus. What's that blood type? O positive. And then for our screen, this is gel, so you only have one reading. So you can record that as gel, IAT, AHG, whatever you want to name it. It's the same test. If you have an ortho, so the difference between tube screens and gel screens, notice it doesn't say it on this, but it says it on the vial. Does it say this is suspension on the cell screens? That says 2 to 3% suspension. So in tube, your reagent cells are 2 to 3% suspension. If you're using gel on this ortho, which ortho is not the only manufacturer that makes gel screens. Grifols has a gel screen. Biorad has a gel screen. They are 0.8% suspension. So it's much less red cells that are being added. So basically, if you see that you have a 0.8 suspension of red cells, you know that's in gel. So if someone forgets to write gel, you know if they recorded it on this, that it's in gel. So you see here, they have a positive reaction for cell 1 and cell 3. So what would the interpretation for the screen be? Yes, positive. It's not asking you what the antibody is, it's asking, is there an antibody present? Yes, it is positive. So O-pos, screen positive. Okay, and we can start washing. Everyone should be incubated for 15 minutes. So after the 15 minutes, you're going to do 3 washes. So that is, you're filling it up 3/4 full with saline, spinning for 1 minute, and dumping the saline, adding more saline, and then so the whole point is making sure that we're washing the cells. So you can see that the cells have tank down to the bottom. So if there was hemolysis here, if you notice that one cell was much pinker than the other cells, you would know cell 3 has hemolysis. When you're washing, the point is to wash the RBCs. So you don't want to mix anything that's free of the RBCs. So you want to kind of aggressively wash. It's going to decrease the amount that you have to mix with the transfer pipette. So you don't want to stick it in the tube, but you want to hit it as hard as possible. So all your cells are suspended. You don't need to use the transfer pipette to mix, because you mixed everything up already. And then you'll spin, and then you'll just don't add more saline. A total of 3 times. On your 3rd time, you want to take a Ken wire, and then so after you've dumped, you want to block as much as you can out, because you want those RBCs to be as dry as possible when you add your anti-RBC. Okay.
Doing all at the same time.
Yeah.
Wash that and
Yeah, so you're gonna, yeah, add saline, all of them, spin. Add more, yeah.
How many minutes is the?
1 minute. Walking in 1 minute. And then when you guys are washing, I'm gonna talk. So gel testing. Carry on, like, don't stop what you're doing to listen to me. But add your saline, start spinning, and then I'll keep talking. So if you dump right now, you're dumping everything into the trash. So that's why I say it's important to understand what's happening. So yeah, if you add saline and then immediately dump them out, your cells are in that saline. So you just dumped your whole reaction out. So then if you did that, you'd have to start over and do your 15-minute incubation again. Does that make sense to everyone? It happens in every class. So after you spin and you get your tubes out, you should see red cells in the bottom of your tube. So you can do ABOs and gels as well, which we will do in a minute. So the ABO cards, this is just one version. There's lots of different versions, but we're going to use the ortho cards. They come with the antisera already in the vial, the tube, or well, the gel card. Because antisera is pretty stable. It does not come with the RBCs because RBCs are not stable. You can see after 15 minutes, they had already settled. So you have to add your reagent RBCs to your ABO cards. And then you add your patient cells to your forward type, just like you do in tube, and then your patient plasma to your cells. So I will pass around some cards so you see some examples. So it doesn't come labeled, it just, the last two are buffered. You add A1 and B cells, so even though they're not labeled, that's what's in the last two. So you can tell by looking at the forward and the reverse if they match. You can just take a look and then pass it around. Tell me what these gel cards, what type does this look like? So we didn't talk about what the reactions look like in the gels. I don't know if you guys watched that lecture yet, but what they look like, if they're negative, that means there's no agglutination. So when you spin, all the cells are going to spin down. So negative looks like all the way to the bottom. If they're positive, it means all the cells are agglutinated. So they're not going to go through the gel matrix. They're going to just sit on top. So basically any red cells that are not all the way in the bottom is a positive reaction, as well as hemolysis. Positive reaction. That's reviewing. So usually for the gel reactions, the grades are, if it's not a solid 4+, what all the cells are at the top. If most of the cells are at the top, but some are coming down, that would be a 3+. For 2+, if there's basically cells all throughout the gel, that's a 2+. And then if there's a lot of cells at the bottom, but there's some going up, that's a 2+. I mean, 1+, sorry. Mixed field, for mixed field, you would have cells at the top and at the bottom. You have a clear 4+ and 0. I would call that 3+.
And then these would be considered, right? That would be before.
And then this one's these are pretty good. But I don't know, they're designed, that would have been at work once.
Are they more where it had their?
No, it's just the way the those
Oh, I see, okay. Negative.
and these were for us.
You didn't say that.
What is it?
Okay, thank you.
Yeah. That's what.
Uh-huh.
I don't have to take my.
Oh.
I'm on the hall, I'm my phone. Okay. Oh, no possibility. No problem.
Like, whoop.
Yeah. You know what? And I got. They got you good already.
And then you're gonna fly it, it's one number two fly.
So talking about AHG, there is poly AHG and there is mono AHG. So if you have anti-IgG, it only has anti-IgG in it. So poly AHG has anti-IgG and anti-complement in it. So different labs will have different policies. Some use poly, some use anti-IgG. Complement is usually detected in cold antibodies, which are usually not clinically significant. So we only use anti-IgG because we don't really want to see reactions to complement. So we will use just anti-IgG, should be in your wrap. There might be both, but it will say on the vial what is in it. So that has both. IgG and that one is good and they're ready to be.
Did you add them?
They're rocket.
Yeah, this is the fit already.
No, they're not.
That's good. I thought,
Oh,
yeah.
So. Okay, you have the company, you don't even have to. You don't know. So dump it and.
Yeah.
And then, I will, Friday, this time.
But here, and,
Yeah.
yeah, we have to read it.
You can create everything else.
So.
What it sounds like to me. And then it's all like.
It's not an FDA rule that these have to be green. It's just green because it's nice to see, and lots of people like to see it. Because you know you dropped it if you see it's green. They do make clear too, but most people like the green. So these two, and
So this is what I'm looking at, what it is, feel confident, but it- Let me ask her.
well, this one is, it's still monoclonal. But
Did we add the headline speak and? Oh, not. Well, really, terrible. I guess we. Different manufacturers, but all. Of course.
When I was.
And usually. It's a.
I don't think it's so. Yes, that was you. Yes, in the video, it looks like. Yeah. Yeah, it doesn't.
So after you add your 2 drops of anti-IgG, you're gonna click it to mix, spin for 20 seconds, and then read.
I think I'm using this one here too. Yeah, this one's small, it's personality. No, you use just the regular, just the regular. So, that is one actually simple line. Yeah, I mean, yeah. It's one, you don't read. No. So, and you have one, right, one, that is-
We're gonna set up reaction on general. And see what that looks like. Yeah, you're just. Before you spend anything, you wanna just mix it up, make sure that everything is mixed. Okay, good, and then make sure everything is figured out. If you have any in your back. So check cells are not very stable, because they are cells that have been sensitized to IgG. If cells are sensitized to IgG, eventually they're gonna dissociate. So the check cells, even if you don't see a positive reaction with your check cells, it's because these check cells are expired. So the cells are starting to hemolyze, the IgG is breaking free. So it's normal to see a negative reaction in class. In the real world, if you're using in-date IgG or check cells, you would have to see a positive reaction. But if you see a negative reaction, it's not because you did anything wrong, it's just because the cells are expired. And yeah, it should be very weak. Yeah, and like the complement check cells, like probably ever working. Like it's like you see a female stagnant on the bottom. So there's not check cells in every rack. The check cells are called IgG sensitized red blood cells. Okay, so when you see a negative reaction after AHP, you're gonna add 1 drop of your sensitized red cells. So you can see, even before they expire, they start hemolyzing because they're sensitized. So these are darker than the normal agent cell. So you can use them, but if you see a negative reaction, obviously you probably didn't do anything wrong. It's just the cells are not really great. You ready? Yeah. You can still add a check that you should see some illumination. What is it? Yeah, that's the 2. That's yeah.
That would be a 0. Oh, okay. So after you spin and read your AHP, you're adding 1 drop of check cells. Yeah. Can you repeat that? You're reading your AHP, favorite reactions, add 1 drop of check cells. Spin and read.
Yeah, you,
Yeah, it's not a blue connection, right? I would do 8 for I don't think that can look like this. It's gonna be 8 or N. So you want to go to screen on your program. Okay, because if you don't have the anagram, it's not really gonna tell you anything. It's not. Okay, so on an anagram, you list your immediate spend and then AHG read, and then you write check after you do the check. Okay. That one. What is the next? Oh, there. So for all the workshops, whenever you submit the study questions at the back of the lab manual, you also need to upload any of your lab worksheets where you're recording your results from today. So your RH worksheet and then your anagram for the screen, you'll upload with your study questions. Okay. Has everyone read their AHP? Has everyone added their check cells?
no.
Do you see anything with the check cells? You did?
Yeah.
So any reaction with the check cells, if it's weak, 1+, you're just writing check. So you added
AT phonetic. And then this will be considered like it's.
So when you break this up, those are your cells, it's not monomerase. So the red is the red.
So it looks like you just. Lost yourself in this one.
One of them, just when I put it up, it wasn't something out.
Another, one, I think I told you, I'm in some.
No.
So
Like,
like, so, tomorrow, it's when I'm gonna put you read that, so you can tell whenever you tell the computer. Do you want to mix it or you? Okay, now we're gonna go to the back and do our Dell stuff. Did everyone read Pixel?
No, yeah, yes.
You read your text sounds. Oh, not one. It was lighter. We were 8%. So 8%. Was supposed to be accepted. Everything ready. You see?
Apologize. You are. I know it's out there on it.
We have a gel. Gel will come with this gold foil on top. So an ABO card. This whole card will be used on one patient. They record the patient names. And then up here, if you add anything to it, or you're going to add A1 cells, do so.
The last two are done.
They're just buffered, so we're gonna have. these up. So if you're doing, this card is an anti-IgG card, so it has the anti-IgG in the gel. If you're doing screens, you can use one card for 2 patients, but you have 6. So you would just record whatever patient, and then 1, 2, 3 for your screen cells. So these have already been tested. So you can pass that around. And then there's also Anacera cards. So if you want to do like mass screening of your donors, or if you have a patient that has an anti-little e and like screening all the units you have in your fridge for little e, then you can add, basically each well is gonna be 1 patient or 1 donor. So this has anti-E in there, so the screen has anti-IgG. These have anti-E already in them, so you're gonna add your patient suspension. And positive, the patient's is little e positive, they'll have a positive reaction. A negative, they'll have a negative reaction. So, we're doing, and so for any of these gel things, there are instruments that don't want to for you. You're in a big blood bank, can't fight that with a fight. You have 200 patients coming in every day. You're gonna put on your patient sample, and the machine is going to find out everything for you. But, you're in a reference lab, or if you're in a tiny little lab and only get 1 patient at a time, a lot of times it's faster just to find out yourself than to QC and instrument to do what they do. So, gel is kind of the opposite of tube, and tube, you always want to drop your clear reagents first, and gel, you cannot do that, so you have to drop your RBCs first. 50 microliters of reagent A1 cells to the well. The A1. 50 microliters, and you just want to make sure that there's also people who go on. And then I'm going to add, so ortho has this neat little pipettes, because basically anything in gel that is going to be either 4.5, 25, or 50 microliter. So this one changes like this. So we add 12.5 of our patient RBCs to the fluid clip. So it's anti-A, anti-B, anti-B, and then control. So the control is just controlling, but there's no spontaneous elimination in your patient cells. So that is why it's called a D control. That's kind of an old thing though. Not really a D control when you get ABL. If your patient is AB positive, you need to have a control to make sure that the patient is not just spontaneously eliminated. So when we have an AB patient, AB positive patient, you must have a control. Then I need to add my patient plasma to my last 2 models. The nice thing about this is it is a beer pipette. So if you're doing a screen or a panel,
after you add all your panel cells to each row, you just take your plasma and just type that along. Is it like come to a stop? Then how do you know you? That's the right amount. Well, you just push down. There's only one push down. That's what I'm saying. You use that for a stop and that's the amount. But you still have some in your tip, right? So these are all patient platform, that's so you fill up your tip. And then each touchdown is whatever. So then AVO carbs, they're immediate spin, so you just immediately spin it down. If you're doing a screen, it's the same. If you're doing it with food, you want to make it 37 degrees. So the gel stations, there's manual gel stations. So they have an incubator up here that you would incubate in for 37 minutes. But if you're doing immediate spin, then you will start here. It's your tubes. These are preset with the correct RPMs and the correct times. So you can see you can't change anything. Just hit start. And that will spin for 10 minutes. And while it's spinning, we'll talk about other stuff. So one of the virtual labs that you will be doing is on RH phenotyping. So Anacera is just reagent with a known antibody in it. So whatever antigen you're looking to see if your patient is positive or negative for, you're going to find the Anacera for them. So like for the RH phenotyping, you have the anti-B, which we already did. It's part of the normal types. There's anti-big C, anti-little c, big E, and little e is the standard RH phenotype. So, from the genetics lecture, the important things that you need to know are zygosity. So meaning, is this patient or is this cell homozygous or heterozygous? So to know that, you need to know which antigens are pairs. So in the RH system, big C and little c are pairs, meaning either you have in this one location on the chromosome, you have a nucleotide that will tell you if you are positive for big C or little c. So if one of my chromosomes says I'm big C positive, my other chromosome says I'm little c positive, what does that make me? Yes. So the reason we care about that in blood banking is when we do antibody rule outs, when we get to antibody ID, which will be our next workshop, for most things, we want to use homozygous cells because some antibodies, if they're new, like developing and you have a low titer, you might not see it if you only have a heterozygous cell, which talk about dosage on the next screen. Because if I'm big C and little c, it means I have basically half the amount of both of these that someone that would be homozygous would have. So if I have an anti-big C in my patients, they're going to react 4+ with the homozygous, but they might only react 2+ with the heterozygous. or if I'm developing an antibody and I don't have a lot of it yet, I might get 0 and then only 2+ with homozygous. So you want to make sure you don't miss a weak antibody, because it can still cause a transfusion reaction in your patient, even if it's weak. And you also need to know zygosity when you're selecting anasera controls. So, talk about dosage first. So this is just what we said. Some antigens do show zygosity. So RH, all the RH antigens, the KID antigens, Duffy antigens, MNS. So JKA and B are in the KID system. So just kidding. Remember, KID. Something I always remember from when I was learning blood banking. KIDs are notorious for delayed hemolytic transfusion reactions. So you get blood, it looks like the patient's doing okay. Just kidding, they're having a transfusion reaction. Delayed. That's just how I remember it. So keep that for board review. So yes, how do we get zygosity? It depends on your parents, who you're inheriting from. So if mom is heterozygous, dad is heterozygous, you can have kids that are homozygous and heterozygous. And we care about this when we get to anasera QC. When we're doing anasera QC, you want to control your anasera control every day of use. So you might only use anti-little c whenever you have a patient that has an anti-C or anti-little c. What most places will do, they'll keep a log of anasera control so that you can see, go to that log and see, okay, has anti-C been QC today? If it hasn't been QC today, then you need to run controls with your patient. Because every day you want to make sure that that anasera is still working. So for your negative control, basically you will use panel or screen cells to test to make sure your anasera is working. So for example, we are testing anti-big C. So we want to find, I'm looking at the ortho panel VSS 400. I want to find a cell that is negative for big C. What cell is negative for big C? 2 or 3. I can use 2 or 3 for my negative cell. Does everyone see that? So for your positive control, this is very important. And this is something that people do not understand, like every year. It's very important that you select a cell that is heterozygous, not homozygous, because we want to make sure that that anasera can detect the weakest expression of the antigen. We don't want to call a heterozygous patient negative if they're not negative. So how do we know if something is heterozygous or not? So you need to know the allele pairs, basically. So all these are pairs. So when you're looking at a cell, you need to know which ones go together. Because basically, if you're looking for a heterozygous, so big K and little k are allelic pairs. If both are positive, they are heterozygous for both big K and little k. If you're looking at cell 2 up here, it is homozygous for little k. So if you're QCing your big K anasera For your negative control, you can select cell 1 or cell 2, negative for big K. And you would select cell 3 because it is heterozygous expression of big K. Does that make sense?
Say that again, it's heterozygous for big K,
Yes.
okay, even though both big K and little k are positive?
That's how you know it's heterozygous. If it's big K positive and little k negative, it would be homozygous for K.
Okay, well, we don't know if it's big K or little k at that point, but it's homozygous for one of them if it's a plus and a minus.
Well, you know, because this is telling you what antigen you're looking at.
Okay, so whichever one is the positive, that would be. Okay, so if it's positive for little k, then it's a little k homozygote, right?
Let's look at Duffy. So looking at Duffy, A and B are allelic pairs. And then same with JK. We'll look at JK because there's no heterozygous on Duffy. So the JKA and JKB are allelic pairs. So these are co-dominant expression. Most blood group systems are co-dominant. So if you have both genes, you will display both antigens. And that's like a typical board question is most blood group antigens display co-dominant genes or co-dominant inheritance. So if I am QCing anti-JKB, let me zoom in on this. JKB, what do I want to use for my negative control? What was that? The third cell is positive for JKB. So I do not want to use that for my negative control. So 1 is negative for JKB. So I want to use that for my negative control. For my positive control, I want to use the cell that's going to have the weakest expression of JKB. That's going to be one that is heterozygous. So cell 3, because they're JKA negative, they're homozygous. So they're going to have extra JKB. So I want to use cell 2 because they're going to have less JKB. So looking back at this, sorry, zygosity. So this sister and this sister are both JKA positive. This sister has a JKA and a JKB. So they're going to have half as much JKB on their cell as this person. So we want to QC with this person, not with the homozygous. We want to detect the weakest expression when we're QCing our anosera. What questions do you have? I know this is really hard for people to understand.
So for purposes of QC, is it always the weaker one that you want to look at?
Yes, because you're proving that that anosera can detect the expression.
Okay, okay.
Does that make sense? Why you would want to do that? It's harder to detect weak expression than it is to detect full expression. So this makes a little click whenever it's done spinning. I'll let you know it's done. So, I'm gonna pass this around. Keep it to yourself what you think it is. So write down what you think it is and if there's an ABO discrepancy.
That's just my granddaughter, answer for all, if I see them, you want to look at that. So there's some positive reaction.
Exactly.
Do you want to take a break or do you want to finish?
I want to be finished.
So, yeah, I only have a couple more slides to go over. And then if you want to do some more tube ABOs, you can. Or if you don't want to, you don't have to. We will do more next. Each workshop will add something. So this time we did ABOs and screens. The next workshop will be focused on antibody ID. So we'll do more ABOs and then we'll do antibody ID. We'll also go over RH because RH is complicated. There's a lot going on there. And it's really important that you understand the terminology for RH, which it takes a little bit to sink in. So it's very easy to be overwhelmed in this class. It's a lot of information. So don't feel alone or like you're not getting it. It's a lot of information. So that's why I recommend if you can read any of the lectures or listen to any of the lectures before the workshop, and then the workshop will go over the main concepts, because some things you just have to hear multiple times before it clicks in. So if you read a lecture, listen to a lecture, and then hear it again and again, maybe at some point it'll click. I'm trying to highlight the things that helped me click things in. Everyone doesn't learn the same thing, so in the same way. But hopefully just by repetition, hearing it different ways, it'll make sense eventually.
All the future labs are posted now, so we can start what like.
All the virtual labs are posted. So if you want to go crazy on the virtual lab and get them all done next weekend, go for it. And
when
same,
will you have the like the week before the physical lab, you'll have them up or?
the workshops, I have the lab manuals up. But like these PowerPoints I'm making
As you go,
to kind of,
you'll have. the
based on
protocol,
homework,
so-
what I think needs to be gone over a little bit more. But the lab manuals are all posted. The modules and the lecture portion are all posted too, like all the homework is posted. So if you want to go faster, you can go faster. But keep in mind, midterm is only module 1 through 6. So if you want to just do 1 through 6 and just hang out until the midterm.
Wait, did you give me that one? Oh, wait, yeah. I'm sorry. What is there? Here?
Yes. So how do you know which one you're looking at?
Exactly. I didn't even know there was.
So the cards have what kind of card is on it. So it'll say ABO card or anti-IgG card or Anacera card. So there's an Anacera card and then there's an ABO card.
I didn't get it yet. That's all my question they got. What should I have? Yeah.
For anyone interested, the boosters are available at Walgreens now, the new booster that has the Omicron variant in it. It's available for all age groups.
Who's there for what?
Okay, did everyone see the ABO card?
No.
Not yet.
Okay, back to QC. So we have our controls. You want to make sure a negative and positive is tested each day of use. So it's not each tech doing it. So if the midnight shift QC'd anti-big C, and then I do a patient at 11 a.m., I don't need to do QC again. It's already been done for the day. It's up to your facility to determine what each day of use means. If you're a 24/7 lab, most places just start at midnight to make it a clear. So then you don't have to calculate, okay. Some things have time on them, some don't. So some controls are good for 24 hours, but anesthesia QC, usually you just write the date. So it starts at midnight. So before you do a patient, you want to check and make sure that it is QC'd or QC it with your patient. And why do we want to type our patient to see if they're positive or negative for an antigen? It is used, so anytime you do an antibody ID, you want to confirm that that patient is negative for that antigen, because if we go back to Landsteiner's law, if an antigen is present, the antibody must be absent from the plasma. So if I have an anti-big C, I cannot be big C positive. I should not be making an antibody if I am positive for that antigen. You don't want your body to attack itself. So a really nice thing about phenotyping your patient is you can use that basically as a rule out when you're doing antibodies IVs. So in the hospital, you don't typically have, so you guys haven't seen me pass around some panel. Take one of these and pass it. So panels are basically the same as screens. It's just more cells. Instead of testing with 3, there's different panels depending on the manufacturer. Some are 10 cell panels, 20 cell, there's 16, there's 12. Totally depends on the manufacturer. But basically it's a bigger screen with different donors. And if it's a good manufacturer, they're going to make sure that they have good rule out cells. So you don't want every cell to be positive for JKB or every cell to be positive for JKA, because if your patient has one of those antibodies, everything's going to be positive and you can't rule anything else out. So you want to have a good combination of antigens in your panel. And these are just examples of what panels may look like just so you can see it. I'm not going to do anything with these. The other thing I like people to make notice of on panel or screen anagrams, depending on the manufacturer, they'll give you different information. And this is good for you to have during your exam too. So for example, one of these. This one, it tells you what the phenotype of these donors are. So if you have a test question that says little r person will have what antigens, you have your answer right here. Little r means D negative, big C, big E negative, and little c, little e positive. And it tells you for R1, R1 and R2, R2 as well. So these can help you if you get stuck. And some of them just have nice information on them randomly. Just depends on the manufacturer, but make sure you read all the fine print because it could be useful in your antibody ID and it's useful on that one.
What do you do for each one?
So you'll learn about that in other studies. It's going to be like- we will talk about that. Okay, everyone see the ABO card?
Yeah.
What is it? Okay, and is there ABO discrepancy? No, okay. What is an ABO discrepancy? Yes. I also have an ABO discrepancy lecture. If you'd like to go over it, then you can stay and go over it, or you can leave. I'll leave it up to you guys. Okay. So ruling in, ruling out, this is just an introduction. Because we're just talking about screen right now. Next workshop, we're going to talk about antibody ID. Basically, you want to be 95% sure in whatever you're saying. If you're ruling something in or you're ruling something out, you want to be pretty certain that you are correct. Different labs will use different rules. So some will say you have to have 3 positive reactions, but they don't say if they have to be on heterozygous or homozygous cells. Or some may say you can have 1 homozygous cell and 2 heterozygous cells. Some are harder to find. If you're a reference lab, you probably have a million panels to choose from, but hospital labs usually do not. Panels are very expensive and you have to change them every month because red cells degrade quickly. So hospitals, because they're not doing that many antibody IVs, the majority of your patients are going to be negative. They usually only have 1 or 2 panels. So you might not have 3 homozygous cells to rule in or rule out an antibody. So again, I'll say it a million times, always follow your facility's SOPs. So for our class, the antibody rules are listed on the exam, they're listed in the lab, they're listed all over the place. But to make things really simple, this set of antibodies can be ruled out without regard to the zygosity. What does that mean? That means you can use homozygous or heterozygous. As long as you have a positive cell, you can rule out that that antibody is not present. Because these typically do not show dosage, so you're not worried about missing a weak reacting antibody. So the ones that do show dosage, you want a homozygous cell to rule out on. So that is all these. And these antigens, they're not super common antigens. So if you do not have them ruled out in what you're given on a assignment or test, you don't have to rule them out. These are low frequency antibodies, so it's very uncommon to see these. And you only need 1 cell in this class. If you can rule it out with 1 cell, so for these with heterozygous or homozygous, with these with 1 homozygous cell, it's ruled out. You don't need to do more cells. It's not going to be like that in the real world. It's going to be either 2 or 3 cells to rule things in or out. But to make things simple for this class, you only need 1. Okay. Make sure you turn in your sheets that you recorded on today with your study questions. And I would recommend doing your study questions as soon as possible, because we just talked about all this now, it's fresh in your brain, just get it over with. So then you don't forget. Because the longer you wait, you're gonna be like, I remember something about that, but I don't really remember it, so just get it over with.
How do we turn these in? Is it like a fillable form or?
Those you will need to upload as a PDF. So you can either scan it in on a printer, or there's apps on the phone called PDF scan where you can take a picture and it'll make it into a PDF.
Yeah, oh my god, I love every end of the slide is the.
You'll see my class a lot. This is my little crazy. So I'm gonna go ahead and start going over the ABO discrepancies. So there's options. If you want to do another ABO, I have more tubes. Does anyone want more practice with the tube ABO? You guys want to do them?
Yes.
So while we're doing that, I'll talk about ABO discrepancies. You can dump it. So you can, if you're doing another ABO, you can record it on the same ABO worksheet. Just make sure that you record the correct donor ID. It can be done. So the tubes that you already tested, the donor tubes, they are plastic tubes, so they can go in any of the biohazard bins. All the glass tubes need to go into a sharps bin. So if you're done with testing, you can go ahead and start putting things up. Your saline tubes and transfer pipettes in the drawer down there, your racks up there. And there is the reagent, blood bank reagent bridge over there. If you're done with your rack, you can put that in the fridge. And your centrifuges and illumination viewers can go up. Okay, so ABO discrepancies. Does everyone feel good about what you're looking for when you're doing the ABO test? What the forward type is telling you, what the reverse type is telling you. Okay. So like I mentioned before, the 4 different discrepancies are missing or extra in your cells or forward typing, and then missing or extra in your reverse or plasma. So for your ABO type, you can just use the ABO lab manual for your directions, since I have this PowerPoint up.
Just start it, please.
Keep those boxes, and we'll fill it up with one of the filled full boxes. So we don't have good like storage things, so we'll just borrow from another box. And the ABO discrepancy homework that you guys did, it's kind of to get you thinking about what's happening, because it's kind of reverse of what you'll actually see. Normally you'll get the reaction and you have to make a call of what's going on. I think everyone's turned that in already.
Spin it again. I could just switch this one out. Or spin it?
Yeah, that's fine.
But we already have.
Okay, first discrepancy. So the first thing you want to think about is, what is the problem? So that is there an extra reaction or is there a missing reaction? And the other thing you want to keep in mind whenever you're looking at ABO discrepancies, you're assuming the stronger reaction is the correct reaction. So your anti-A is the strongest reaction. So you think this is probably an A. But the discrepancy is that we have a weak reaction with our B cells. So what are the causes for weak reverse reactions? So this is a problem with the patient's antibodies. It seems like they're not making enough antibodies if you're getting a weak reaction. Yes, so we can see that in immunocompromised patients or in older patients. So it's perfectly normal if you have a 96-year-old to have weak reverse. It's still called a ABO discrepancy, but we can be pretty sure it's due to the age. So that's why looking at the patient demographics is very helpful. Looking at the diagnosis, looking at the age, looking at pregnancy status. So how you would resolve this, does anyone know?
Well, compromise, you want to look at history or whatever the demographics is, and normal.
You would still, so if it's like an older patient or immunocompromised, because you have a weak reaction. So you would incubate for longer, either incubate for 15 minutes at room temperature or incubate for 15 minutes at 4 degrees. Because your antibodies are IgM, so they're going to be cold loving antibodies. So they're going to react stronger at 4 degrees. So you incubate. So that's when you would record on your ABO worksheet, whatever you did. So 15 minutes at 4 degrees. So ABO discrepancy worksheets are normally to have longer sections. So if the original type looks weird, it'll have immediate spin or that does not be room temp 15 minutes or 4 degrees 15 minutes. So if you incubate at 15 minutes room temp and it goes up to 2+, you could keep going, incubate for 15 minutes at 4 degrees. That might bring it up to 3+. It's not like we really want to go crazy on this because you have a cause for the discrepancy. But usually the first time you see a patient, you want to do as much as you can on the investigation to make sure you're confident in whatever you're saying. So second discrepancy. Do we have an extra or missing reaction? In the forward or reverse? Yes. So what can cause extra reactions in the forward? So again, you want to go with your stronger reactions. So 0 and 4+ are pretty strong reactions. So you think, according to the reverse, this looks like an A patient. So what could be causing this extra reaction with anti-B?
It's an acquired Z caused by gram-negative bacilli, like within the gut.
Yes, thank you. This is a very common board exam. Acquired B. What's the resolution? Well, the cause, you're going to look at the diagnosis. If it's any kind of colon cancer or any kind of intestinal bacteria, it's going to resolve once the infection is cleared. So there's really nothing you can do about it while it's happening. But you can say, you would write this up. But again, this is not something we would see in current day. This is always a board exam question, but because we use monoclonal antibodies now, we don't really detect this. So if this happened current day, you would likely not have a reaction with anti-B. This isn't really a problem anymore at all, but you need to know it for board exam. Because back in the day, we would use polyclonal, and polyclonal will have some unspecific reactions. So you would pick this up when you're using polyclonal reagents. Next one. So extra or missing. And this is a picture of what, if you do an ABO on a slide, I don't know if you've ever done this like in high school, or if you go to donate, sometimes they'll do a slide type. They just put a drop of your blood on a slide. It's like a quick and dirty ABO. If you buy like ABO kits on like Amazon, usually it's done like this. So you just put a drop of blood on a slide, put a drop of antisera, and then you take a stick and mix it together. And then if it agglutinates, that's positive. And then if it doesn't agglutinate, it's negative. So extra or missing? Okay, so we have an extra reaction, but also have a weak reaction with our anti-A. So,
It's a strong reaction on the correct.
right. So we have strong anti-B antibodies and weak anti-A1 antibodies. So when we have a weaker reaction with anti-A, we're thinking it's probably a subgroup of A. So subgroups of A's can make antibodies to A1. Some subgroups make A1 antibodies, some subgroups do not. And it doesn't mean if you're A2, so 20% of A people are A2. All A2s do not make anti-A's, but some A2s do. So if you have an anti-A1, it's normal for A2 phenotypes to have it or not to have it. So that's in the ABO discrepancy homework. That's why plus or minus was a answer. So for example, an A2, it could be plus or minus. It's common both ways. They may or may not. Okay, so when we have A subgroups, the extra things we have in our rec that we can use to help us is we have A2 cells, because since A2 is so common, 20%, it's pretty common. You can test their plasma with A2 cells and see if they have an A2 antibody. So if they're just a normal A2 phenotype, they should not have antibody to A2. So they might have a 1+ to A1 cells, but a 0 with A2. And then we also have anti-A1 lectin, which may or may not be in your reagent rack now. So that is, so I don't know if you guys have seen anything about lectins yet. Lectins are basically antisera made from like seeds. And it's kind of weird because it's like, how did people know? Like, did they just test a bunch of seed, milk, water with a bunch of cells and see what happened? Because we have a lot of different lectins that we know about. So it's kind of cool. But anyway, A1 lectin will react with A1 phenotypes, but it will not react with A2 phenotypes. So that is how you can confirm that this would be an A2 subgroup with an anti-A1. And when you have an A2 subgroup, you want to report if they do have anti-A1, because it can go either way. But A2 subgroups, you don't want to give them A blood, even though they are A, because they can make anti-A1. So you give A2 subgroups O blood. Mhm. I see you're thinking about it.
You said 18 can actually take.
This is an A2 that has made an anti-A1. So that's why you don't want to give A red blood cells if they need a transfusion, because they have an antibody to A1. So most or 80% of A's are A1. So if you give an A2 and A1 and they have an antibody, they're going to start attacking those cells. So they're not going to have a good transfusion. Okay, next one, extra or missing.
Or I guess missing, can you have the missing, or you get the extra.
So
Oh,
if you're looking at the forward, it looks like an O. You're looking at the back, what does it look like?
okay.
Yeah. So you're missing a reaction with your A1 cells. So a good thing to do, you're thinking this might be a subgroup at this point. Anti-A, B is an antisera that should be in your rack. So the thing you need to know about ABO antisera, anti-A and anti-B are IgM. That's why we can do immediate spin testing for IgM. IgM are pentamere. You're familiar with what those look like, IgM? So they're so big, and because they have 5 basically antibodies put together, they can agglutinate on their own. They don't need the anti-IgG to cross-link. They can cross-link all by themselves. So that's why you can see positive reactions and immediate spin. Because your anti-A and your anti-B are IgM antibodies. But O blood groups also make an IgG that is anti-A, B. It can react with both A or B antigens. And that is important when you get into O mothers, because if they have A or B fetus, IgG can cross the placenta, IgM cannot because it's too big. But that anti-A, B can cross the placenta and start attacking the baby's cells. But usually ABO, hemolytic disease of the newborn, is very mild and it doesn't really cause the baby much distress. So that is when you see babies that need to be under the bilirubin lamp. They're under the lamp and then it goes away. It's nothing long-term. They just have a little bit of jaundice. So anyway, what we would do with this one, because we want to see if it's a subgroup, is we would test with anti-A, B. And usually, if it's a subgroup, an A or B subgroup, the A comma B is a little bit stronger and it would show a reaction. So we have a reaction. It's most likely a weak A. And we think it's an A because they do have anti-B. So we don't think it's a B subgroup. We think it's an A subgroup.
I have a question. If you look at that, like, why couldn't it be O?
Well, anti-A comma B would not react with an O.
I don't know, I mean, like, if you were looking at that, just like that previous slide. Like, if you look
Because
at
it wouldn't be normal to make only anti-A or only anti-B. If it's an O, they should make them in pretty comparable numbers. Unless there's something, some subgroup going on. So AX is a subgroup that will react very weakly. Sometimes it'll react with anti-A comma B, sometimes it will not. Some of the very, very weak subgroups, you have to absorb and elute to actually get reactions. But a really easy thing to do to resolve ABO discrepancies is genotype the patient. But a lot of hospitals don't want to pay for genotyping. So that's, yeah. Sometimes you just have to use what you have. Okay. Next one. Missing or extra reaction. So again, when you're doing these, good thing for you to do while you're getting the hang of ABOs is look at the forward and say, what does the forward type tell me? And then look at the reverse, like look at them separately. So the reverse type looks like yeah. And then the forward, it looks like a B2, but you have mixed field. So remember what I told you earlier today. What does mixed field tell you? You have two cell populations. Two cell populations. That can be caused by different things. It can be caused by transfusion. It can be caused by a chimera. It can be caused by stem cell transplants. If you're converting, if I'm an A and then I get an O transplant, I could have two different cell types floating around at the same time. Chimeras are when there are two embryos and they merge together and you can have different stem cells in one person. So that happens. It's very rare, but it does happen. And it can also be caused from a traumatic delivery. A mother could have fetus blood circulating in her blood, so she could have her type and the baby's type floating around. So lots of different reasons for mixed field. The other thing you do want to remember for a board exam, the three subgroups, A3 and B3, are mixed field. So in this case, it looks like it's a B3. If you look at transfusion history, there's no transfusion history. Then you're thinking this is a B3 subgroup. But just remember, 3 goes with mixed field. No, 3 are the only mixed field reactions in subgroups. If you see mixed field, you're thinking 3. So the only way to resolve this would be with genotyping. There's nothing else you can do serologically to prove that this is a B3 until you genotype. Okay. Now, do we have extra or missing? Look at your strongest reactions. So now, when you have extra reactions like this, there's a few different causes. So what you want to do is you want to run a screen and auto control. So you want to, let me see. So in one patient, we run the screen and the auto control. The screen cells are positive and the auto control is negative. So it looks like they have a cold allo antibody. In the second patient, we run the screen and the auto control and both are positive. So it looks like we have a cold auto antibody. So when you're looking at antibodies to determine if it's an allo antibody or an auto, you want to look at your auto control. Remember, you're testing your patient's plasma against your patient's red cells. If they're reacting, they have an auto antibody. Auto antibodies don't rule out that there are allo antibodies present. You can have both, which is a big pain in the butt. This will be on many exams. So make sure you're clear on what the auto control is telling you. So it's also important you know that the A and B reagent cells are pooled as opposed to screen cells. For screen cells, each vial has one donor in it. So you know exactly what the phenotype is. It's on the anagram. For the A1 cells and the B cells, it's a bunch of different A1 donors and a bunch of different B donors pooled together. So if you do have a cold allo antibody, like anti-M, you're going to get positive reactions like this, because there are some donors in there that are positive for M. So does anyone know how you would resolve that? Because if they have an anti-M, you can't test with the reagent cells.
You can warm it up.
You could pre-warm and see. But, so cold antibodies are typically IgM. So if you warm it up and it doesn't go away, what you can do is you can take a donor unit that is labeled A1 and a donor unit that is labeled B, phenotype those donors for M. If you have done an antibody workup and determined it is an anti-M causing this discrepancy, phenotype some donors, you find a B donor that's M negative and an A donor that's M negative. And you're basically treating those donor cells as your reagent cells. So you can prove that this AB patient does not have A1 antibody or B antibody. Does that make sense? That was a lot.
That's something that was like done at one blood or something.
At Life South, yes, or One Blood. Yes, that's what they do. They send off to a reference lab, which reference labs are normally at blood centers. Yes, although they don't have to. This is something that a hospital could do on their own. It's really about who you have and if they know what they're doing. Because they should have A and B donors on their shelves at all times, and they should have anti-M antisera that they can type those donors for, prove they're negative, and then use them. But if they don't have a SOP for that procedure, then yeah, they would send it out. Most hospital labs will send out ABO discrepancies. Unless you're like Shan. Shan's pretty much does everything on their own. Except platelet stuff. Okay, next one. What is going on here? missing or extra?
Missing?
Yes. So at this point, we have no idea what's going on. So what I would do at this point, one thing, I would redo everything. Like, did I add reagent to all my tubes? Was it, did I mess up? Make sure I add anisera to my tubes and plasma to my tubes. And then if I'm still getting this reaction, I'm going to incubate everything at room temperature for 15 minutes and read it. If I still get nothing, then I'm going to incubate for 15 minutes at 4 degrees and read it. You should get some reaction after 15 minutes at 4 degrees to point you in the direction of what's going on.
So the incubation like at 30 minutes, you say a good sample.
I've never seen anything like this happen.
Okay.
Like, it's not going to be common at all that you would get no reaction at all. You would probably see weak reactions if something was happening. If you got a really, really bad sample that was completely diluted with like saline when they drew the sample, and you didn't have a lot of plasma in there, you might see weaker reactions in your reverse, but it shouldn't be, there's at no point that there's going to be no plasma in a tube. So this would be most likely human error. Something was not added. Okay, so now we have a gel. So this is this is not ortho gel. This is Gripples, I believe. And they make different ABO cards. Some have reverse, some don't have reverse. Some have multiple D anisera, which we'll talk about RH a lot more in the next workshop. So what is the problem here? What does this look like? What? It has mixed field. It does. So when you see mixed field, yes, thank you. A3, we're thinking A3. And that's another thing that can be plus or minus. When you test A3 with anti-A comma B, usually you get mixed field 2. So that would be a good thing to look at. So it would be good to think A3 if you did not see this AB. But since the AB is positive, what you would be thinking, and one thing you want to look at where the most cells are. So not very many negative cells. So what I would be thinking, because you don't have the reverse here, so that's not helping you. The reverse would tell you what's going on. But what I would think is this is an A person that was transfused 1 unit of O negative cells during a need for uncross match blood. So they came in a car accident or something, and they gave them a unit of O negative. So mostly A cells of their own, but some O negative cells. Or O net positive, because you don't see any negative cells in your D. But yeah. Okay, here's a very poor picture, but you can still clearly see the reactions. So what's going on here? So one thing, is this an ABO discrepancy?
Negative.
Does the forward match the reverse?
Yeah, it does.
But you have mixed, right?
Yeah, so it's not an ABO discrepancy. Remember, D is not part of ABO. So no ABO discrepancy, but you do have a mixed population. So this, a negative person could have got transfused A positive cells or vice versa.
Okay.
So this is likely due to either they were transfused or It depends on what this antisera is. If this is a monoclonal antisera, then it's looking at one clone. Some D are D blends. So in your rack, if you look at your anti-D, some will say D blend, some will say monoclonal, some will say polyclonal. So we'll talk about this a lot more next week. I love the RH system, so I will talk about it all day if you ask me to. But the really confusing thing about D, when you're looking at an RBC, D is a multi-pass protein. So the D antigen will do like this across the cell membrane. So there are weak D phenotypes and there are partial D phenotypes. So this would be a D positive. And then if you have no D, you're D negative. So there are some phenotypes that are weak D, meaning they react weaker with anti-D. So weak D is a quantitative difference. You still have all of the D antigen, you just have less of it. So you react weaker. So because you have all of the D antigen, you will not make an anti-D. You have D. There's nothing to make an antibody to. Then there's partial D. So you may react 2+ only. You're missing epitopes of D. So if each piece of D that sticks out is an epitope, so we'll call this epitope 1, 2, 3, 4, 5, 6, and then we have a partial D that has 1, 4, and 9. They can make an anti-D because if they get transfused a regular D, they're not going to recognize 2, 3, 5, whatever, and they're going to make an antibody to those epitopes. So remember, go back to immunology. Antigens can have lots of epitopes. So the epitope is where the antibody will bind. So if you have a monoclonal anti-D, it could just be specific to epitope 3. So if you have one of the partial Ds, it's not going to detect a partial D. But if you have a blend of D, it's going to have anti-1, anti-3, and these are not real names. There are specific names for these anti. This is just for simplicity's sake. We're going to talk about this more next week or next session. But partial Ds are qualitative. They can make anti-Ds. Weak Ds are quantitative. They cannot make anti-D. So anyway, if you saw a discrepancy like this, you would look at transfusion history. It's likely due to a transfusion.
And that reaction is like immediate, like you had the one question that they had the transfusion. Like
Well,
the
for
day
the cells,
before.
the cells would be immediate because you have those donor cells floating around. If they're making an antibody to the donor cells, that's not gonna be immediate. It's gonna take some time for those
They would
antibodies.
Show up on a test that they had like the day before, had a transfusion.
But again, it depends on the size. If you have a big person that gets 1 unit, you might not see any mixed field. If you have a baby that gets a transfusion, it might be very obvious. So it depends on the size of the transfusion, the size of the person. And in a tube, it's going to be much harder to see. And gel, it's much more obvious when you have a mixed cell.
So do you always usually do both or?
It just depends on the facility. And this is our last one. And it's kind of hard to see because of the background. It's hard to take pictures of gels. So missing reaction or weak reaction. Or missing or extra. So forward reaction, super strong, looks like an AB positive. So again, this is an example, an AB positive where you have to have a control. Your control is telling you you're not having spontaneous agglutination in your cells. Your control must be negative if you have an AB positive person. If this control is positive too, we don't trust these reactions. Does that make sense? That means in your control, your cells are just agglutinating spontaneously. It's not because it's a true positive reaction with anti-B or anti-B. So you just have some extra reactions in your reverse typing, which this could be caused by an AB patient being transfused with O plasma. They're taking some of those, or they have some anti-A and anti-B in their system from that plasma that was transfused. Or this can also be caused, have you guys seen Rouleau in your hematology courses? So Rouleau will show in your reverse type, if the patient has multiple myeloma or Waldenstrom's macroglobulinemia, one of the diseases that makes you make a lot of plasma proteins, can make your cells really sticky. It makes your plasma sticky pretty much. So you can have extra reactions that's due to those plasma proteins, can cause problems in your reverse type. So this might be Rouleau. If you suspected this was Rouleau, you would switch to tube and view it in the microscope to see if you have the stack of coins appearance. If you see that, to prove that it's Rouleau, you have to spin down your reaction, your reverse reaction again. And then before shaking it, you're going to pull the plasma off the cells and do a saline replacement. So if you pull up 2 drops of plasma, you're going to replace it with 2 drops of saline, mix it up, spin it again. If you read that reaction after the saline replacement, if it's a negative reaction, you have proven that it was Rouleau causing the agglutination.
Can you explain how to fill this up?
Yes.
So if it's negative, compared to below, whenever you after you do the saline replacement, you do 2 drops of saline, 2 drops of saline along with 2 drops of serum, and if it's negative,
No, you don't add your saline. So if you do your regular reverse with 2 drops of your plasma with the reagent cells, and you see Rouleau, what you think is Rouleau, then you're going to spin it down again. You're going to pull off the plasma. So when you spin it down, instead of shaking it back up to read it, spin it, pull off the plasma, leave the red cells there, add saline. So if it's a true antibody that's bound to the cells, when you pull off the plasma, the antibody is still going to be on the cells. When you pull off the plasma and add the saline and then you see no reaction, you're proving that there's no antibodies bound to those cells. We can draw it out. So it makes sense. So this is how these are recorded. So phase, so you did an immediate spin. Your bottom row is your auto control. And then you chose LIS or PEG and read at AHG, and you record your reactions. So you should all have negative reactions for AHG. So you're just going to have 0, check, 0, check.
We don't have any.
So the other thing, you will see other labs. This is a side conversation. I don't want to confuse you. You're asking how to fill this out. So you're going to have a row for immediate spin. You're going to have your AHG, and then you're going to document whatever you chose, LIS or PEG. You're going to just write that above. And then, for example, if you chose PEG, your 37-degree. You use PEG, the only thing you can record at 37 is hemolysis. So you would record no hemolysis, no hemolysis, no hemolysis. And then at PEG, you would document 0, 0, 0. And then for all 0 reactions, you must add check cells. So you would document check, 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 but you check to make sure they're all working. So you can record like that. That's how I prefer to record check cells, because it takes up less space. Some labs will add another row and just name it check cells. And then they'll actually record a reaction for check cells, like 1+, 1+, 1+. I don't like that. It just takes up extra space, and you don't really need to know what reaction it is. That's why I prefer just to do those. Yes, I checked, and everything's good. But both of these is something you might see. Both acceptable ways to document check cells. Okay, so rouleau. So what is happening? This is your reagent A1 cell. And in rouleau, you have plasma that has a lot of proteins in it. It's extra proteins that are causing rouleau, which is basically sticky proteins making the RBC stick together. It's a problem because it makes you think there's agglutination when there really isn't. It's just stickiness. So you're adding your patient plasma, and this is the rouleau stickiness. And then as a side, this would be reagent A1 RBC. So when I'm showing something, I like to show you like what's going on in one thing and what's going on in the other thing, so you know the difference in what's happening. So this would be in like a multiple myeloma patient that has extra proteins causing rouleau. And I cannot spell rouleau off the top of my head. I'm so sorry. There's too many vowels. As opposed to a B patient that actually has anti-A. So in this one, you're actually going to have, because anti-A is a anti or a IgM, which is a pentamer. So it's going to make all your cells really stick together. So if you see some agglutination, what you're going to do, spin your cells back down. So if you have true antibodies, whenever you spin down and take off the plasma, you are removing all these, all the sticky plasma. they won't stay stuck to the cell because it's just sticky. If you have a true IgM, they are bound to the A1. So after you remove the plasma and then you add back saline, all the stickiness has been removed. So you're just going to get a clean negative reaction. Your RBC is clean. If you have a true anti-A, these are not going to leave if you take away the plasma. You can take away extra IgM, but they're still going to be bound. So you're still going to have a positive reaction. If it's a true antibody, you're going to have a negative reaction if it's the protein caused by rouleau or rouleau caused by the protein. You look so confused.
I'll have to read about it because isn't it something to do with you do the list for the zeta potential, and that zeta potential is what prevents, like, there's a bunch of negative charges around that cell, and if you're putting in the list, that's going to.
So that's different
Oh,
because in the ABO, you're not using anything like that. That is the function of list, low ionic saline. But in rouleau, it's just the plasma that you're removing for the saline replacement.
and because it's not actually bound. So what about, why not do an elution? Like to look and see like what's underneath.
Because it's a 20-second immediate spin.
Uh,
An elution is like an hour-long process. So you want to make things as simple as possible. An elution costs money too, because it's a kit. No reason to do an elution. Because there's nothing sticking to it. If you did an elution on this, there'd be nothing there. Because there's no antibody present. An elution is just going to elute off antibodies.
Yeah, anything, on the cell. Okay. But then that'll be obvious in a question because everything will be positive. Like,
All your plasma reactions would be positive. So that's why we do a positive and auto control too. Because yeah, the auto control should be positive too if it's rouleau causing the reactions. So your A cells, your B cells, and your auto control should all be showing rouleau. Because it's the plasma, it's not the cells. It doesn't make any sense. It doesn't make a difference what cell you're adding, because you're going to see it on your screen cells too. Any tube that has your patient plasma in it is going to show them.
it won't be just like one showing another one.
No, and that's another thing. When you have unspecific reactions, you're thinking it's an unspecific protein or something causing it. Because if it's reacting with all cells equally, it's not a reaction to an actual antigen. So it's not an antibody reaction. It's a non-specific protein reaction.
That was good. Are you gonna post this lecture?
Yes, and this is a extremely long. Yeah,
That's super helpful, what you're doing with the drawing.